Hello.
Hi. Emily and Jaden.
Hello.
Hello.
Hi.
Hi, Emily. All right.
How you doing?
I'm okay. You know, busy. I didn't expect it to be this. Sure, you guys feel the same way.
Oh yeah.
Yeah. Yeah, I just, forgot something I was supposed to do by 5 o'clock. So basically, it's just applying for money. So I guess I won't be getting that money.
I forgot to apply for unemployment this week, so
So yeah, mine was like spent time helping people get ready for online because I spent a lot of time over the break helping North Harris, Houston North, and a lot of time. So they were gonna, compensate people, but I just lost track of time today and.
what were you helping with?
I have,
Like reorganizing curriculum?
making their course. No, they don't have any online material. So like basically for 14-7, a course for them. Because like you can use some of my course, but not all of my course because their class was an 8-week class, so they hadn't even started yet. It was from the beginning.
Oh, wow.
Hey, Abby. Hey, Susie. Anyway, so it was like hundreds of hours. Well, maybe not hundreds, but at least like 50 hours.
Jaden.
Yeah.
So it's annoying, but I'm just glad to have a job.
That's good.
Yeah. So, does anyone have any questions or things they want to definitely cover, first of all?
Chemiostasis. I think that's what it's called. I'd like to kind of go over that, review that a little bit. It's
Stasis?
chemiostasis. The chemiosis? Word? I don't remember what it's called.
What concept?
It's relating to cellular respiration, I believe.
Okay.
I remember working on it.
The osmosis?
Yes, that's the one.
Okay, got it. Okay, there we go. So we can, I want to do some comparisons in cellular respiration. Why don't we start with that? Yeah, let's just start with photosynthesis, cellular respiration kind of side by side, which can be helpful because then you can see the similarities and differences, and then you'll also know the expectations of how much detail I want you to know.
Okay.
Basically, a lot of it's like, what's the input? I think I have it on one of the modules. It's like, what's the input? What's the output? Why are we doing it? I'm not too big into numbers. Like, I don't need you to know how many of this is formed, how many of that is formed.
With cellular respiration, there's a lot of different reactions where there's like one that will have like how much ATP does this reaction yield? Is that what you're talking about?
Yeah. I just need you to know it's a lot or a little bit. Is it a lot of ATP or is it a little bit of ATP?
Okay.
And how does that matter, or why does that matter? So, I can do a number of things.
I believe
So you guys pretty much just want me to review stuff. Is that correct?
Yes.
just general review would be great.
Okay. So, And I have a thing that I did this morning, with my the 2-hour session this morning, and it went pretty well. And I did a lot of review stuff. So that's still processing. It takes a few hours to process the recording. I'll put that up too if you want to. You can. I think you could scan through it and see anything else that you want to do as well.
Okay.
Okay. So. Let's do this. All right. Okay, can you see that drawing window?
Yes.
Great. Okay, so one thing is that I put it up, and okay, there's already the lecture about it, and I put up another reminder this morning about it, which is the overall process that's cycling from inputs of photosynthesis to the outputs of photosynthesis, which become the inputs of cellular respiration and the outputs of cellular respiration. So that cycle is really, really important. And if you can understand that, then you're be able to apply it, then you're well on your way to being able to answer pretty much any question I ask about cellular respiration photosynthesis. It's this one, this type of deal here with the inputs and the outputs of photosynthesis and what converts, and then the inputs, the outputs of cellular respiration, and what processes do the conversions. And there's like an extensive, I think it's called chemical cycling lecture on that. And then this is another one I just did this morning to go over and remind you. So that's really important. So I'm going to look the little bits of that together. So you should be able to know, I'll just do this. People tend to do better with photosynthesis and cellular respiration. I'll just let you know that. So don't overstudy the photosynthesis and forget about the cellular respiration. I'm going to go for this here. This is the basic inputs and outputs of the whole photosynthesis process. Okay, so basic input is water. Water goes into the light reactions, and what comes out? Oxygen. What else is involved in light reactions? Not surprisingly. The reason I'm making these green is because they're the ones that depend on the absorption of light by the pigments. These are do it in blue, so it goes with the light reactions. Okay. And there's many, many, many thylakoids, and that's what these oblong things are. They're the stacks of membrane that's highly folded inside the chloroplast, and that's what this whole thing is. And then I have a extensive lecture on this, so I'm not going to do it crazy. I'm just going to remind you here. Okay, we've got CO2 coming in, and we have the carbon compound coming out. G3P initially gets converted to glucose. Plants gets converted to sucrose, could be stored as starch, could be stored as used as cellulose, any of those things. This is the Calvin cycle. And I'd say, okay, why do we do the light reactions? Really? The whole thing of the light reactions is you're making molecules to run the Calvin cycle.
So are the green circles, is that just representing the different chemical reactions that occur during the light part, or is that?
No, those are the thylakoid membranes, the stacks of membranes within the chloroplast.
Okay. And then on the right, when you represented the Calvin cycle, that's just happening in the stroma, right?
Yes. And I mean, I always do it as a circle, but you're right. It's just reactions. The enzyme binds the two substrates and the reaction happens. Then they get released. Then another enzyme will bind them and they get released. So you're right. It's not literally a circle.
Okay.
The cycle part of it is just that you have a molecule you start with, and at the end of the whole process, you end up with that molecule again. So you have a new starter, and it just keeps making that starter molecule over and over and over.
Okay.
That's the cycle part, yes. And these green things are the thylakoid membranes. They're the stacks with the pigments and the photosystem and all that stuff in there. So they have the photosystems, the ETC, ATP synthase, and we'll look at them a little bit.
Wait, I thought the ETC, I thought that occurred on the membrane of the what you call it? That's the electron transport chain, right?
Mm-hmm. Yep, and it's in both cellular respiration and photosynthesis.
Oh, okay. Okay.
Mm-hmm. It's the same, different complexes for the electron transport chain. Same exact concept. I'll show you that in a minute, comparing those. Same exact ATP synthase, same outcome of making ATP. It's just the ATP made here with the light reactions is strictly used to build the sugar in the Calvin cycle.
Within the chloroplast, it's only thylakoids with the reactions happening inside it, but it's mainly just like a vessel for thylakoids.
So it has the stroma, which is the empty space inside of there. It's fluid filled, and then stacks and stacks of, they're like coins, but coins with like a hollow part inside, or pitas, maybe pitas stacked on top of each other that are all connected. So there's lots of those inside, but there's also lots of empty space too. And the empty space is where all the enzymes are, all the molecules that are involved in the Calvin cycle. Because everything requires enzymes out there.
It looks like there's a few more people who are still trying to join.
They can jump in. I mean, they shouldn't have a problem with that, I hope. Are they say they're stuck out?
Justin said it's just not working for him. I'm gonna help him figure it out.
Which link did you guys use?
I went to D2L.
Okay, tell them to use that. Maybe it's
Okay.
messed up.
To try his Lone Star email, not the D2L email, because that's the one that works for me every time.
Yeah, that's the ones that I have. The ones I send the direct invitation to are your Lone Star emails.
The one that worked for me was that I just used the app on my computer, and then I go to like the number of the meeting and I copied and pasted because it didn't work for me when I used the link. I think that's what he's trying to do. So.
Okay, so if he has the email on his My Lone Star, that link should work. And I think, I mean, on D2L, I believe on the homepage, I tried to put a link on there. But in any case, for the thylakoid stacks, Abby, you were asking about that. So here's your double membraned chloroplast. And when we say membrane, we mean phospholipid bilayer. Same exact thing that we talked about with the cell membrane, the phosphate head with the two tails, hydrophobic tails, hydrophilic heads, they face each other. So you've got two of those bilayers going here. And then you have a bunch of same phospholipids in these stacks, or making these stacks, which you can see are interconnected here. Here's what it looks like with this is like a microscope cut through view. Here and they're connected. So those are the thylakoids. And in the stroma, ribosomes are out there making protein. That's where the little bits of DNA that the chloroplasts have that code for certain proteins that are chloroplast specific. That is Calvin cycles out there, all those reactions. And then the thylakoids are where all the light reaction stuff happens, capturing light, electron transport chain, hydrogen ions get concentrated right in this very small space here. And then the ATP synthase is involved that way too.
Is the ETC within the the thylakoids, does it produce like a large amount of ATP like the one in the mitochondria does?
Anything, yeah, anything with ATP synthase is going to produce a significant amount of ATP.
Okay.
If it's doing ATP without ATP synthase, like it does in the citric acid cycle and the glycolysis, just a little ATP. But anything with ATP synthase makes a lot. Now, it's not enough to run the whole cell for the chloroplast because the chloroplast uses this ATP specifically for building the sugar. That way the cell doesn't have to make ATP separately, like with its mitochondria to build its sugars. It makes it within the chloroplast, and so like the whole thing happens within there. Okay, so ATP, oh, let me finish this part. What's the point? What's the point of the Calvin cycle? The whole point of it is to that's how I think of it. Embed electron. Oh, you can't see that. Switch back. Sharing. iPad. Okay. So the whole point of the Calvin cycle is to embed electrons in a carbon compound. Part over here. Embed electrons in a carbon compound. And why do we want electrons? Because electrons, basically, electrons in a bond, which is where they are, equals potential energy. So the more electrons that get embedded in there, the more potential energy in the carbon compound, which means like when it gets broken back down in cellular respiration, you have a lot of energy to extract from it. So you need ATP to be able to build. Why? Building is an anabolic process. That's from chapter 6 stuff. Anabolic process, so it requires energy. The energy initially comes from the light and then ATP, and eventually it's going to end up in the carbon compound. Molecule. So besides the ATP, that's the energy to build the compound, anabolic, what else do we need? Electrons. Building a carbon compound and embed electrons. So that is the other product of light reactions. Like I said over here, why do we do the light reactions? To make stuff to run the Calvin cycle. So the NADPH is just a molecule and it carries electrons. Basically, it's taking electrons from water initially, transferring them, and then putting them in the sugar. And then it's just a cycle because after you drop off the electrons, now it's empty. Plus sign means that it has lost its electrons. So it's just recycling back to pick up more. And the ATP has been broken to use its energy, and so it comes back as ATP. Why do you need water? That's where the electrons keep coming into the system. So if we were to try to track electrons, we track them from water into the NADPH, and then ultimately electrons will be in the carbon compound. So that's the electron pathway. So that's photosynthesis overall. The opposite process is cellular respiration. It has a lot of the same elements, but it's well, stuff's happening.
Do we need to know anything about like bacterial cellular respiration or fungal respiration, or is it just going to be animals and plant cells?
So the thing you'd want to know about bacteria is that they can do these processes. You just don't use the mitochondria at chloroplast. The whole thing is the bacteria that turned into the mitochondria and chloroplast in eukaryotes. So they do it just with their cell membrane. The membrane is very, very, very folded. Bacteria that can do these processes. And that folded membrane is what they use to do that, which is not surprising because that folded membrane is what ends up being the folded membrane in the chloroplast and the mitochondria anyway.
Okay, so
anything else besides that? They have ATP synthesis as well. They can do, some of them do fermentation, which I will briefly.
like yeast.
So yeast is a fungi. Like the bacteria that produce the sour yogurt taste. It's lactic acid, lactate.
Hey.
Hi.
Here is the really folded membrane I'm talking about. So here is the type of bacteria that became a chloroplast, got engulfed, didn't get digested, so got eaten, didn't get digested, ended up forming this symbiotic relationship with the cell it lived in. But you can see how folded the membrane is. And this type of photosynthetic bacteria, this is what became the chloroplast. All these thylakoid membranes inside. And this type of folded membrane is what the bacteria that can do cellular respiration use. And that way long time ago, I'm talking about, billions of years ago, 2 billion years ago, something, maybe 1.6, something like that, really long time. But this type of bacteria with the folded membrane got engulfed, didn't get digested, ended up forming the mitochondria. So it wouldn't be surprising that the ATP synthase and the electron transport chain and all that stuff is on these membranes. The membranes are now on the inside of those organelles. Anything else besides that for bacteria? Not really.
Anything that we need to know specifically about bacteria for cellular respiration or
just that they do it the same way, except for they don't use the membrane.
Okay.
They don't use the organelle, they use their cell membrane and the highly folded structures to make this concentration gradients.
Okay.
Where's Dad? And when is he gonna get back?
Carson.
Okay. So that's that. Let us.
Just letting you know you're not on mute. Just letting you know.
I know these kids. Oh, hold on. These kids won't ever leave me alone. We've been together far too long.
Well, you're a teacher and their mom, so.
Okay.
Not alone, Abby. You're not alone.
Carson, stop. Stop.
Okay, so this is the cell. And this is grossly enlarged, not even close to proportional. But if I don't draw it this big, I can't get stuff in. Okay, great. So that's the mitochondria. You want to know the different parts of that. That's pretty straightforward and what happens in each part. And so then we just draw the very, I'm going to draw this. I'll just draw the most basic parts here of the process. All right, out in the cytosol, we have the glycolysis. Call that number 1. Then we have, in that process, we have glucose to pyruvate. Transition there. Let's just track the molecules for the moment. Pyruvate goes inside the mitochondria. Process 2A happens. Come on. I call it oxidation of pyruvate. And I put a little short video up about oxidation reduction if you need a reminder about that. Basically, oxidation means loss of electrons. Oil. Pyruvate is being stripped of electrons. At the end of that, the pyruvate ends up being acetyl-CoA. Why do we make that? Because it's really reactive. So we can go into the cycle. Call this 2B. That's the citric acid cycle. And an important goes into this. And then the third part here, we don't have any carbons going on here. But all this other stuff, the whole point of it was to get electrons for this. I'm drawing embedded in that membrane. Electron transport chain. And that ATP synthase. ACE, remember, means enzyme. So the ATP synthase is an ATP making enzyme. It's an enzyme ACE that synthesizes ATP. A lot of it. So that's step number 3 here. Those two together. Oxidative. Not it. Oxidation. Oxidative, and that just means oxygen needs oxygen. Oxidative. Pho, like phosphate. Phosphorylation. So it's the ATP making using oxygen, or involving not directly oxygen, but dependent on oxygen. Oxygen dependent ATP making by phosphorylating. So that's like overall. Can I move to another slide? You just have to unmute yourself and say, wait.
Continue.
Make it. Okay. So then if we just look at carbons. They're not in the forms they're in. I mean, like they're molecules. I'm just a carbon. That's really all we need to do. It's basically like track the electrons, track the carbons to a very basic degree. So this is your glucose, C6. That's why there's 6 carbons. Okay, so glucose in glycolysis gets broken. Glyco meaning glucose, lysis meaning breaking. I'm just going to draw step 1. You see that on the other picture, so then you can keep track of that. So it doesn't get totally wild all on the same picture. Okay, so glycolysis breaks the sugar. Why? Part of that is so that it can extract electrons. So here's a really important concept. Break a bond, extract electrons. It's like the name of the game in cellular respiration, basically. Break the bonds, extract electrons. That's basically what the whole point of these steps up to the electron transport chain. And I'll say usually or often make a little ATP. Not always, but often it makes a little ATP. And why are we extracting electrons? Send them to the electron transport chain. And that's because the electron transport chain is what part of that whole oxidative phosphorylation to make ATP. Like the whole point is to make ATP. How are you going to do that? Using electrons. How are you going to get those electrons? And then you just have to have a shuttle to get the electrons out of the one molecule and into the electron transport chain. And that's what the NADH, NAD plus, that's the 2 versions of the molecule with and without electrons. Okay. So what do? We broke a bond. So that means we can extract some electrons. Break a bond, extract electrons. So how are we going to, what are we going to do with them? We're going to put them in the empty cab. So this cab has the light on because the plus, or the plus is like without the minus, the negative electrons. So I just think of this as a shuttle. When it has the plus sign, it's like the cab's empty and ready for business, which means the electrons can get put into it. So it gets some negatives. And then it's going to fill that up. And now it has the little passenger of 2 electrons and a hydrogen. So now it's full. It's just the shuttle. We track 2 electrons at a time. So what I would do is, as you're studying tonight, I would just tell yourself the story of the electrons. Okay, the electrons start in glucose, and then glucose bond gets broken, and the electrons get put into NAD+. And then that's sent to the electron transport chain. Then we've got pyruvate, and pyruvate gets broken. And so like that's how I would do it. You just tell a story. Tell the story of the electrons. Okay, so break a bond, extract electrons. And then I also said usually you make a little ATP, which indeed is what happens here. So ADP, and then it becomes ATP. It's substrate level phosphorylation. All that means is that the phosphate's on another molecule. It's not like floating around by itself. It's stuck to something and gets transferred to ADP. I don't really care that much. All I need you to know is that it makes a little ATP, not a lot, because it's not using ATP synthase. Okay, you know that means ATP. There we go. Okay. That can go out and do some work in the cell. And where the electrons go in the electron transport chain always go to the E to C, the electron transport chain. Or where do they come? NADH. Where do you put NADH, electron transport chain? Okay. So that's the pyruvate. And then pyruvate carbon get broken off. And that's one place that you get CO2. So in our bodies, breathing out CO2, where did it come from? The food. Not just glucose. You can you don't have to just eat glucose to be able to survive, right? You can use other molecules at different points in this process. But where is carbon coming? You're breathing out right now, all that's from food molecules that you ate, or your body tissues where you're storing atoms and molecules. Okay, CO2 gets popped off. Why? Because we break a bond. Okay, so we broke a bond. And what does that mean? It means we can extract electrons. Same thing over and over. So again, coming with NAD plus, picks up some electrons, NADH, and that goes to the electron transport chain. And then you add on this unstable atom molecule. It's very large. It's called coenzyme A. Why? Because it makes it unstable. So like, why do you do glycolysis? To start breaking it down and extracting electrons. Why does it do processing pyruvate? It does that because you can extract some electrons and molecule reactive. This is the acetyl-CoA. That means unstable, very reactive. Because otherwise it's not going to do the reactions. Unstable bond right there. So then we take that CoA, acetyl-CoA. Acetyl is just those two carbon groups. And that gets added into the citric acid cycle.
I have a quick question.
Sure.
I have heard acetyl pronounced acetyl. Is that correct? Like aluminum versus aluminium.
Yeah, acetyl-CoA or acetyl-CoA. I hear both. Just like you hear pyruvate and pyruvate, it's like slightly different. Both of them are. We'll use pyruvate and pyruvate. Yeah.
Answers my question. Thank you.
Acetyl group or acetyl group. I mean, because it's a group, you'll talk about in organic chemistry.
Mm-hmm.
I mean, technically with English rules, it should be acetyl, because acetyl is weird because you have the accent on the Y, which is Y right at the end of the word. But I mean, that I also have heard it both ways.
Mm-hmm. Okay.
But you say acetylcholine receptor, it's the same thing. Dictionary says acetyl, no, acetyl and acetyl. Acetyl and acetyl. It says both. Dictionary says both. So the CoA pops in there because it's reactive, and basically you pop off the CoA, stick the electrons on. I mean, stick the carbons on. And this is another key point. One key point was that you break a bond, extract electrons. Another key point is, and I'll erase this in a second, but and it's sort of vague, but this is the idea. So you add a phosphate group. This is one of the ways ATP works. And also the CoA, the coenzyme A, it's the same idea. So what do? You add a phosphate group, or you can add coenzyme A in this specific case to make a molecule unstable, which also means reactive. So it's so big, it's making it very reactive. And then you switch it out. That's what I think of it as, at least. You switch it out, the phosphate or the CoA, for whatever you actually want. So in this case, we actually want to switch it out for this 4-carbon thing. Oxaloacetate, I don't care, you know that. So the 4-carbon starter molecule. That's what we're switching it out for. In the Calvin cycle, you're switching it out for electrons. Do that in a second. Okay. So now we've got a carbon compound. The whole point of this citric acid cycle, the entire thing, is to extract electrons. That's the point. Extract electrons. So how do we do that? We break a bond. And so what does that mean when we break a bond? That means we're popping off CO2s this time. And this is part of the thing that I don't need you to know numbers, but I do need you to know. Break a bond, release CO2 as a byproduct. What does that mean? We can extract electrons. And I'll draw this in a second bigger. Should I just do it now? Well, let's do it now. Okay. Do it now. I want this restart. There we go. Okay. Starter. Very unstable acetyl-CoA. Yes, this happens twice because there's 2 pyruvates, whatever. It's not important. So there's your 4 carbons, your starter. And by pops off, I mean it's cleaved off by enzymes. So every step here is catalyzed by enzymes. Nothing just magically happens. There's always protein enzymes that make these things occur. Okay, the extent to which I need you to understand this goes as follows. Let's just do. Stop. Happens once, extract electrons, and which means Ad plus comes in and picks them up, and ADH to the electron transport chain. We're going to do it again. Break off another CO2, which you breathe out. This is what you're breathing out right now as we speak. And that means you can extract some electrons. And again to the electron transport chain. Like I mentioned before, often when you're doing breaking bonds and extracting electrons, you can make some ATP. It's a complicated process here with the GTP and stuff. I don't care that you know that. That is not important to me. Important to me is that you know it just makes a little ATP. No ATP synthase. And that ATP can go do work in the cell. And then we have more rearranging to get back to our starter. Make more NADH. Make a little of this very similar molecule, FADH2. Basically, we're just extracting electrons like crazy, because that's the point. To electron transport chain, and eventually you remake your starter molecule. And that's the citric acid cycle. And it's the opposite point of the Calvin cycle. So what's the point here? Point is to extract electrons from that carbon compound to send to the electron transport chain. And that's like the whole carbon. That's the carbon part. I wanted to, before we do the oxidative phosphorylation, I want to draw like this kind of conceptual version of the Calvin cycle. So when you're looking at it tonight, you can have them right next to each other so you can see like their opposites, basically. Does anyone have any questions about the conceptual nature of this, or like what extent I need you to know it? This is basically why are we doing it? Extract electrons from the carbon compound to send to the ETC. Why carbon dioxide come out? Because you're breaking bonds to extract electrons. Who takes the electrons away? NAD plus, and becomes NADH and takes those to the electron transport chain. Make a little ATP. That's what I need you to know. Okay. Now, switch your brain over to photosynthesis, but it's the cycle in photosynthesis, which is the opposite, embedded electrons. This is the Calvin cycle. Find us. Calvin cycle, part of photosynthesis. Similarities and differences. It's a cycle, so it also starts with a starter molecule. It's a 5-carbon. The point is to build carbon compounds, so that means we need carbon. Where do we get our carbon? CO2. So look at this, it's an input of CO2. Instead of popping it off, you're bringing it in. Okay, so it brings it in. It's an unstable molecule, so briefly it's like that, but pretty soon it goes and breaks down into 2,3 or splits into 2,3 carbon compounds. All right, good. Now we brought a carbon in, so we brought in new carbons. Now what are we going to do? Second part, build a carbon compound. What are we going to do? Embed electrons to make it high energy. A lot of electrons and bonds. So in order to do that, we need to bring in electrons that we made in the light reactions. So we're going to have from the light reactions NADPH. Now it's phosphate P is phosphate, but you can think of as P as in photosynthesis. Before it was NADH, right? Without that. That's going to be our electrons from our light reactions. And remember, we also made the other product in the light reactions, the ATP. That's our energy. That's coming from the light reactions. From the light reactions. Okay. So in order to get the electrons in, this molecule is not reactive. So just like I mentioned with the CoA, what are we going to do? We've got to make it reactive. How are we going to do that? Put a phosphate group on. That's what ATP is used for. So I'm just going to draw one of them because it's more messy. Great, so you got the ATP, added a phosphate group to the carbon compound, makes it reactive. That's the point. The squiggly bond means it's unstable bond. So I'm going to make that reactive there. And really it's reactive right at that carbon. So we're going to switch it out. It says you can switch it out, pop off the phosphate, and it's, you know, it's all happening by enzymes. And what we really want, we don't want the phosphate. We really want is the electrons. And they're coming from NADPH. Let's put them in the sugar. And then it's an empty cab. I'm ready to go get more. Yay, that's what we wanted. Now we've got our product, sugar or a carbon compound with some electrons in it. Good, so that means that we can have one of those exit and be used by the cell. So that is our G3P molecule, the first carbon compound that's made. G3P, aka carbon compound. And the basic idea here is then you rearrange all the rest of the carbons in order to make the starter molecule again. And yeah, you need to go around 3 times with the carbon to count the carbons. I don't care. I don't care about that. If you take biochem, you will learn lots and lots. And by that point, you'll have taken organic. And so it's kind of cool, actually. I really like biochem in that regard. It's like you've taken this basic stuff, then you took organic chemistry, so you'll understand how reactions work. And then you get to look extensively at photosynthesis and cell respiration, but you understand it on like a molecular level. I try to bring a little bit of that in, like with the phosphate. Why are you using ATP? How is that working? And I feel like if you understand that it makes the molecules unstable so they can be reactive, then that's like, okay, what do I need to do? I need to do a reaction. How am I going to make that happen? Oh, that's right. Add the phosphate from ATP. Why am I doing that? I want electrons. Switch out the phosphate for the electrons. Now I've made what I want, carbon compound with electrons. Take one of those out of circulation, the rest of them get rearranged.
Isn't it, with that being said, a little counterintuitive that we take this course before we take organic chemistry or like some sort of?
No, I mean, so usually people are taking this course while they take Chem 1, although honestly, Chem 1 is about balancing equations and stuff like that. I mean, bond-wise, it's useful. But, I mean, a lot of people won't have to take organic. It just depends on if you're going to be a bio major, you will. But
I think
It's,
I ask because the reactions on all of this stuff is really what trips me up every single time.
you can think of it just conceptually, like, here, let me, at least for the, you don't need to know any specific reactions, but here's the idea of how phosphate works. We're trying to go from B by itself and add A onto it to make an AP compound. So A and B are not going to react unless one is unstable. So that's where the phosphate comes in. So here's the phosphate group from ATP. It's an unstable bond because it's very large or very negative. Like all those oxygens just make it very unstable, and it makes this carbon really reactive. And so what do? You pop off the phosphate and switch it out for what you want. Here's an example with just atoms in it. So here, the idea here is that we want this OH group. We want to switch it out for an NH3 group. So how are you going to do that? You got to make this carbon reactive so that you have the reaction there. So what you can do is you put a phosphate on there, make it really reactive and unstable, and then switch out the phosphate for whatever thing you actually want. Pop that on. Now, in our case, for photosynthesis, we don't want an actual we don't want atoms or a little molecular group. We want electrons. But it's the same thing. You add the phosphate group on there, make it reactive, and then you can switch out the phosphate for some electrons in there. So instead of adding an ammonia on or an A on, you just put electrons in. So the idea there is that the ATP, the ATP phosphor, like putting the phosphate from ATP onto something makes it reactive, and then you switch out the phosphate for whatever you want. In the case of the Calvin cycle, for electrons. That's just when they say you need energy. You need energy to build. You need energy to do photosynthesis. And this is why. You make bonds to get electrons in there. Yeah. And it's the opposite, really. Yeah, it's the opposite when you're doing the other one, when you're doing cellular respiration, extracting. In that case, you're breaking carbon bonds to get electrons out. In this case, you're putting electrons into carbon bonds. And if you look at the two cycles we just drew next to each other, I don't have enough room to draw them next to each other in my little screen. But if you look at this and then you put it right next to the one we drew before, the citric acid cycle, you'll see that's basically the opposite. So the other one's making ATP, this one's using ATP. The other one ends up with NADH. This one starts with NADPH. So put electrons in, take electrons out. Make ATP versus use ATP. Make bonds, break bonds. They're just two opposite things. But this is like the extent I need you to understand for how the Calvin cycle works, and that other one's the extent I need you to understand the citric acid cycle. So it's not, hopefully you'll see, it's not like overly complicated with all the enzymes or even the molecule names or different steps. Same with glycolysis. Glycolysis, I just need you to know it splits the glucose, extracts some electrons, makes a little ATP. That's it. You don't need to know all the steps. You don't even need to know all the, yeah, that's what we're looking at. So on this level. To do the last part, with the comparison of the electron transport chains. Let's do that. I can do this on one page. So let's do on this side and the cellular respiration on this side. So both of them use a phospholipid bilayer. It's important because remember, those are regulating what goes across. They're very specific of what goes across. And so that's how this whole thing works, is that it has to be particular of where hydrogen ions can move in particular. Again, very schematic. I draw two lines here to remind you it's a bilayer. And I'm just going to draw this here. And this is very conceptual, so put myself in the room. It's the same thing. I'm just trying to draw it smaller for my, so I can fit it all together. You don't have to erase yours. And if you have to go, I did this morning as well, and hopefully that recording will be ready soon. So you can scan through and find that. Yeah. All right, so this photosynthesis, so we've got the thylakoid. These are those stacks in the middle of the chloroplast. That's what this membrane is. And then the thylakoid space is what's inside of here. So what has to happen? We have to have some input of energy. This is light reaction, so not surprisingly, that's light. The light comes into the system. It's captured by pigments, which are just molecules that absorb light. In the photosystems, photosystem 2, photosystem 1. And this whole thing is about electrons moving. So it's just all about moving electrons, moving electrons, moving electrons. So the first movement here, the light bumps electrons, and they can be transferred to each electron transport chain. So those are 2 electrons. So they go from electron transport chain, fall, And so basically it's just getting, like, as they get transferred, they lose energy a little bit each time. And this is energy that's used to do work. And in this case, its work is to pump hydrogen ions across the membrane, or protons. H plus, same thing. This is the point. Why does it use light? Why does it absorb light and all that? This is why plants need light. So then you've got this concentration gradient where you have a lot of hydrogen ions in the inside, but not a lot on the outside. Outside here is going to be the stroma. And so we know molecules or particles move high to low. So that's what this is going to do. But because it's a bilayer, it can't just go across. Those hydrogen ions, the protons are charged. They can't cross over a phospholipid. So where's the only place they can exit? Through this ATP synthase. ACE enzyme. It's an ATP synthesis. And it relies on the hydrogen ions move through it. It makes it spin, literally spin. And that spinning process changes its shape, and it can make the ATP. And really, in the end, that's what everything is. Everything in the end is shape. Shape and charge and hydrophobic, hydrophilic. That's it. That's what makes everything go. So we have ADP and phosphate. And the ATP synthase makes this, and I need you to know it's a lot of ATP. You don't need to know numbers, just a lot. And in this case, that goes to the Calvin cycle. This is photosynthesis to go build the sugar. Okay, so now we've made ATP, but in order to keep pumping hydrogen ions across, the electrons keep having to get moved. So how do we move, or how do we keep moving electrons? First, you have to have new ones come in. So how do you replenish the electrons? Water. Decide if I want to draw this. Inside or outside. So water is going to drop off the electrons, new ones. Water will drop those off, and when it comes back out, because it's been broken and the electrons get extracted, just like always. So water gets broken, electrons get extracted. There we go. And since water gets broken, what's left? Hydrogens, which just go into the fluid, and oxygens. So you end up making oxygen, and this is the oxygen gas that the plants release. Just a byproduct. So split water, hydrogens end up as part of the fluid, and your oxygen get, two of them get put together, and there's your oxygen, diffuses out. That's where your electrons come from. They keep getting stimulated and moving along. Now, what do you need though? If you want to keep passing, so if you have electrons, you can't get any more until you get rid of them. So you have to have somebody at the end of the chain taking them away. Final electron acceptor, what we often call them. So who's going to take the electrons away? electron carrier. This is photosynthesis, NADP+. And that's going to pick up those electrons. NADPH. Take those little guys away. And it'll go to the Calvin cycle. So we just made 2 products to go to the Calvin cycle. We just saw how those were used. This is where our water comes in. This is where oxygen comes out. So that is photophosphorylation. Phosphorylation, that's this ATP process using light.
Okay, so just a quick question. So from what it's sounding like, a lot of this is, like you said, conceptual. So is that meaning like most of the questions on the test are going to be conceptual, or will you tell us, is there anything that you want to go into with?
A lot of it's like, where do the electrons to replenish the photosystems come from? Like water. What's the energy input for photosynthesis? Light. What's the point of the light reactions? Make NADPH, which has electrons, and ATP, which has energy, to go to the Calvin cycle. Where is the ATP synthase located in photosynthesis and in cellular respiration? In photosynthesis, it's in that thylakoid membrane. Cellular respiration, see, it's in the inner mitochondrial membrane. Why does it matter that all these are phospholipid bilayers? Because that allows this concentration gradient to be built, because it prevents the hydrogen ions from moving across on their own. They can't just get across the membrane. They have to go through the ATP synthase. What is the energy? What is the energy used to make ATP? In the end, it's really the concentration gradient, like the water behind a dam. The hydrogen ions are the water behind the dam. Using ATP synthase, not other processes, stuff like that.
Okay, so like cause and effect.
Why do we breathe? We'll see that in a second. Why do we breathe? There's only one reason. Electron transport chain in mitochondria. Got to take the electrons away. That's it. It's amazing, because you think oxygen must be used for a lot of things, but no, only used for one thing. Plants aren't dependent on us because they do both these processes, so they don't need us, but we do need the oxygen from plants. Or if you're an ocean animal or something, use algae, oxygen. Yeah. You have, you've been working out and lost some weight. How did you get it out of your body? You breathed it out as CO2. We just saw that that's how you break that carbon compound down. And where does the carbon compound go? CO2. So that's where that went. That sort of stuff. Then, of course, remember, too, that there's the cell division and the cancer. You'll need to know the definitions of malignant tumor, benign tumor, what metastasis is, characteristics of cancer cells that make them unusual. And then energy stuff, like where it's endergonic, exergonic, anabolic, catabolic, potential energy versus kinetic energy, how ATP works, and we've already talked about that here. One of the ways is putting phosphates on to make it reactive. Basically, the other way is putting phosphates on to change something's shape. Enzymes are very specific. They work because of their shape. They bind substrates, inhibitors, competitive versus non-competitive. The idea of feedback, So it's like the end product goes back and stops the pathway. It's really great because when you have enough end product, it stops its own pathway. But once you don't have enough product, then the pathway is not stopped. So it makes more product until there's enough, and then it stops the pathway until you run out of product, and then turns back, or it's free again, not to be stopped. So that feedback idea is really important biological concept. And the reason we talk about that is usually it's blocking enzymes, is the product blocks an enzyme in the pathway. And then when there's not enough of it, the enzyme's not blocked, and so it works. Basic idea of fermentation. They do a really nice job in mastering about that, and it's in the book. I mean, I'm not, yeah. I also did a little thing about that this morning if you want to look at that. It's the same as the book, but.
Will we be able to access this video? I had to come in late because my daughter got into paint.
Oh, goodness. Not yogurt colored into paint. That's what my friends do. They're like, we're just making paint out of yogurt. But so this one's probably, I don't know, it takes a few hours for it to get loaded. The one from this morning should be accessible soon. I mean, I had been checking since we were done at 1, so that one will be up. And basically, let's see if I have it. Basically, it's the same thing. Search for recordings. Let's see if it's because this one's. I mean, it takes a few hours. Oh, good. Office hours exam Q&A. Yep. Okay, so it took about 3 hours and 45 minutes to process, but I will share that one immediately. Okay. All right. There's your review office hours, morning on Piazza. Sort of morning.
Thank you.
Okay. I do want to draw this other thing, and and so people have to go. I understand. It's just you'll have that other thing as well. Same thing though in cellular respiration. And I do have a question about like compare mitochondria and chloroplast stuff. So nice comparison here is they both have ETCs. They both have ATP synthase. They make an ATP in large amounts. They use these concentration gradients. They have the cycles happening in the stroma or the mitochondrial matrix. So that'd be something to think about. This one here. We're looking at 2 bilayers, so that's why. Smaller. It's drawn like this. And the only reason I draw this like going around the corner is because it's just easier to fit stuff in. There's not like ATP synthase. It's actually on the bends. It's on the folds of the membrane. But do it this way. Okay. So this is again an ATP synthase. I'm not going to draw the name again, but And this is again an electron transport chain. Different complexes, same idea. No photosystems. So there's a lot of the same things, but it's a lot of it's like sort of opposites, but the same. So here's an opposite. Instead of NADPH taking the electrons away, NADPH, or excuse me, NADH, no, NADH brings it in. So remember, this is cellular respiration. Now we're in the mitochondria. So the matrix is in the inside. In the intermembrane space. So we have NADH with the electrons, drop those electrons off, and leave as NAD+. So you see it's the same thing as dropping electrons off, but in this case, the NADH is dropping them off. In photosynthesis, the NADPH is leaving, taking them away. So we still get electrons into the system. They still fall down the electron transport chain. That still allows us to pump hydrogen ions across. Now, in this case, we're pumping it this way. Why? Because this is the little side, the little area. This is the squishy. Easy to make a concentration gradient when it's very small, folded. High concentration. FADH2 does the same thing. So worry about NADH. Anything that's carrying electrons. And where did all this NADH come from? All those steps we did before. Break a bond, extract electrons, send them to the ETC. That's these things. So NADH from glycolysis, NADH from oxidation of pyruvate, NADH from citric acid cycle. All these end up here. Pump the hydrogen ions across. Awesome. Made this concentration gradient. Awesome. Now we need the hydrogen ions to flow through, which they will. That concentration gradient, potential energy, the water behind the dam. Same thing happens because they go through. The ADP and the phosphate group get converted into ATP. And this is that oxidative phosphorylation. So this.
I have a question.
Uh-huh.
So I guess I'm a little confused. Is this in organisms that can do photosynthesis and cellular respiration, or is it like one or the other?
Any organisms that are doing cellular respiration do this. If they're doing cellular respiration and they are a eukaryote, they will do this in their mitochondria. If they're doing cellular respiration and they are like a bacterium, they'll do this in their folded membrane, their cell membrane. But bacteria do this, animals do this, fungi do this, plants do this, protists do this. Yeah, basically.
So so in animals, because animals don't typically do photosynthesis,
no, none of them do.
What are they doing instead of photosynthesis to pair with the cellular respiration?
Eating.
Okay.
Yeah.
So does eating kind of do this similar process?
So eating process is Chloroplast mitochondria in a plant cell. Plants do both processes. And this is useful too, because I want to show you the comparisons. And I'm just drawing one of each of these electron transport chains in each, but remember there's like millions of them. The membrane is full of them everywhere. I'll explain this in a second. All right, you have the chloroplast in the plant cell. The water comes into the chloroplast. The light comes into the chloroplast. And then light reactions happen. The oxygen comes out of the chloroplast as a result of those light reactions. That's what we just saw with the electron transport chain business. That oxygen, oh, shoot. I have to finish this. One last thing is important. How do we get rid of this at the end? Oh my goodness, this is terrible. Skip this last part. It's very essential. So how do we keep this going? Right now, our electrons are just stuck at the end, and this is what happens when you suffocate. The electrons can't get transferred out of the chain. If they don't get transferred out of the chain, remember, if you have electrons, you can't pick up any new ones. So you've got to get them out of the chain in order to keep it going. And if you can't keep it going, you can't pump hydrogen ions. You can't pump hydrogen ions, you can't make ATP. So we're really in big trouble if that happens. So what happens? What takes it away? That's where your oxygen comes into play. This is the reason you breathe. You need to take the electrons away. So you have oxygen coming in, it picks up those electrons, picks up some hydrogens, and leaves as water. Okay, so those electrons are transferred into oxygen and they leave as water. So we made oxygen before, and now we have oxygen here. So that's why they call oxygen the final electron acceptor, because it is the last one at the end of the chain that takes the electrons away. In photosynthesis, the final electron acceptor is NADP+. Takes it away as NADPH. Oxygen takes it away as H2O. And yes, there'll be 2 waters for every oxygen, because you have to, where's the second oxygen? It just gets held there until there's another water made. Okay, so that is really important because it's got to keep it going. So actually, if you were to block part of this chain, let's say you had some sort of drug that, or carbon monoxide, this is carbon monoxide poisoning, one of the ways it works, is that if it blocks any of these transfer steps, even if you have enough oxygen, it'll look like you suffocated in tissues because the cells die in this way. Why? Because even if the oxygen was there, if you can't pass it to the oxygen, it ends up being equivalent to not having any oxygen at all. What reminded me that I needed to draw this is this part here. Okay, so the oxygen, where does it go? It's going to go over here. the end of that electron transport chain, picks up those electrons, and come back out as water. It does not have to go back to the chloroplast, but that's one of the places it could go. Draw the oxygen here again so that you. Okay. So that's the cycling of water and oxygen. Basically, it's bringing the electrons in here, and it's picking up the electrons at the end. I guess that's better. So water brings the electrons into photosynthesis. Oxygen takes the electrons out of photosynthesis in the form of water. Other part here is the Calvin cycle, citric acid cycle. Oh, what's my pink stuff? Why did I draw this pink stuff? So the pink is your hydrogen ions. This is where hydrogen ion concentration is the pink. You can see it's in the middle of the thylakoid space. This is just what we drew with the last picture. In that thylakoid space, all concentrated there. It's in this intermembrane space. But that's the intermembrane space. Those are like equivalent. They're both small spaces to concentrate hydrogen ions. And then saw this. Both of them have electron transport chains. And this little thing here, both of them have ATP synthases. Both of them are making ATP. In the chloroplast, it's used to build the sugar. In the mitochondria, it goes out to do all the cellular work, like moving and doing active transport and doing anabolic reactions that require building. So then the step here, the carbon compound is going to go into, I'm going to draw it green, glycolysis first. So it doesn't actually, it's not part of the cycle really because it doesn't release any products that go back, but it is part of the cellular respiration. So we do glycolysis, converted, eventually goes into this citric acid cycle. You have CO2 coming from the oxidation of pyruvate. You have CO2 coming from the citric acid cycle. And that CO2 is what goes back into the mitochondria. So this is that same diagram, that circle diagram, and I told you it was important. This is showing that in pictorial format. Where does the ATP come into play? This is an output here. ATP for the cell. So the energy initially came in as this light energy. It got converted, so it came in this light energy, got converted into carbon compound energy, and then eventually ended up in ATP energy bonds to the cell to use. So that's in a plant cell that does both. And if I change and you need me to go back, just someone let me know. It's an animal cell. Okay, so you got your glucose from eating. Either a plant or eating something that ate a plant, because the plants are where the glucose or the carbon compounds come into the system. The reason I say carbon compounds too is because G3P is the first product of cellular respiration, but also proteins and lipids can be used as well. They're used in fed in later into parts of the citric acid cycle, or citric acid cycle. But it's not like you have to only eat glucose, right? And where else would this come from? You could even, eat starch. You can break that down into glucose. You could convert other things to glucose. Glucose is the only thing that goes through glycolysis, though. Everything else gets fed in later. And then in this case, you'd have the glucose coming in. You want to do the whole thing, which is going to be the red color that I've been using for cycles. It's going to come in. Through glycolysis. Not lose anything though, so. And this is where things that don't do photosynthesis get their stuff. Okay, so you got to eat to get the glucose or some other carbon compound. The CO2 comes from breaking down that carbon compound, and you breathe that out. That's how you get rid of the CO2. And then basically, where do you get your O2? You breathe that in, and that comes out as H2O, which you breathe out as water vapor. You know how you can fog up a mirror because it's warm, but also the water vapor. And so basically, instead of cycling around in the cell, you have like an input and then an output. And then we do have the ATP That's the part that we get, just this part. How do we breathe it out? And I mean, it's diffusion, right? So it diffuses out of the mitochondria, diffuses out of the cell, into the bloodstream, fuses through the lung, cell membranes, out the body. Savannah, does that make sense? Or savvy, as it were. Sorry, it's been so long.
Yes, that makes sense. Oh, no. She tried to end the call. This is.
Hey, Professor, you're muted.
Unmute me. Okay, I'm on now. Thank you. So here is the idea of fermentation. And the book has a really pretty picture of this. The whole point, though, is to make ATP without oxygen. So it's in a low oxygen environment. The cell still needs to have ATP. Some cells are small enough where this is fine. Some cells, like our cells, can only run for a little bit on it. So you make ATP. You can do that with glycolysis. Glycolysis doesn't require oxygen. But in order for glycolysis to continue to go, you have to have the inputs. So you need ADP. That's not a problem because the cell's breaking down ATP all the time. So that's no problem. As long as you have some eating part or input, glucose is not a problem. It's this NADP or NAD plus that's the issue of regenerating, because usually that comes from the electron, like when it drops it off at the ETC. But if there's no oxygen, everything gets backed up. There's nowhere for NADH to drop off its electrons, so you get stuck with a lot of NADH and very little NAD plus, which is problematic because then glycolysis can't go. So what'll happen is that the electrons will get transferred either into pyruvate straight, like straight up into pyruvate, and that's what I'm showing here. So those two electrons get put right into pyruvate, make lactic acid, aka lactate, same thing. Pretty efficient because pyruvate's already being made, so the cell just ends up using it as, and it's not doing anything next because no oxygen's there, so it can't do anything in the mitochondria. Everything's backed up. And so what does it do? It has enzymes that will just put the electrons from NADH right into pyruvate. In other cells, pyruvate is first converted into another molecule, and then the electrons go in. I'm not worried about those molecule names. You'll just know that in another process, there's like two steps, and one step produces CO2, and the other step produces the ethanol, and that's what yeast do, which is why they make bread rise, the CO2 part, why they can make beer and wine, the ethanol part. So if you were trying to home brew beer, and you couldn't get it to work, if it wasn't producing any ethanol, you might consider the oxygen levels. If there's too high oxygen, yeast won't do this. Yeast will use the mitochondria, which does not produce ethanol. So you have to keep that yeast in a low oxygen environment. So the yeast is eating the glucose, or the maltose really, malt sugar. Maltose is 2 glucoses. So the yeast break the maltose into 2 glucoses, it eats the glucose, makes pyruvate, some ATP. If there's no oxygen, it reduces the pyruvate to CO2 and ethanol, and then just keeps on doing glycolysis to make ATP so it doesn't die. And yeast are one cell, so plenty for them. Make sure you look at oxidation reduction. I did a little video on it this morning to remind you. It's just moving of electrons. The molecule gets electrons, it's gained electrons, which is Rig, and it's reduced. A molecule loses electrons, oil, oxidation is lost. And you can even look back at your lab 1 with the Benedict's test. That was an oxidation reduction redox reaction. And they also talk about it in chapter 7. Basically everything happening in photosynthesis, cell respiration, is oxidation reduction except ATP. Make that a point. If I ask you a question about oxidation reduction, the answer will not be ATP. ATP just does phosphorylation, dephosphorylation. DT and add that phosphate group on, an inorganic phosphate ion, that's phosphorylation. And then to take off that phosphate is dephosphorylation. So that is, ATP is not moving electrons around. It just gets rid of the phosphate, adds it back on. Gets rid of it, adds it back on. Whereas the, particularly the electron carriers, the other stuff too, all the electron transfer chains, the water, the oxygen, both of those. So picks up 2 electrons. And because it's gained 2 electrons, it's gained the electrons, so that means it's reduced. Or reduction process here, the gaining of those electrons. And when it loses the electrons down here, the oxidation process. Oxidation. So ATP is not moving electrons, just phosphorylation, dephosphorylation. You also have other things besides NAD+. So like, let's say we have water. The water is split and it ends up at oxygen. This is in the light reactions. So if you think of the water gets split and those electrons, remember, go out to the photosystem. transferred into that photosystem, water loses those electrons. So that means water has been lost. Oil. Water loses electrons. So we can say that, how you phrase it, water is oxidized to oxygen. Just say. And then it's the opposite on the other end, right? In the oxidative phosphorylation, the oxygen picks up those electrons at the end of the ETC. Above food. Make it green. Haven't written this that much, so I want to write it for you. Oxidative phosphorylation. Oxygen is going to pick up the 2 electrons at the end of the electron transport chain. It ends up as water with new electrons in it. So in this case, the oxygen is reduced because it's gained electrons. And you always have to have the opposite. So like in the top case, the water is oxidized, but the photosystem is reduced. Because like electrons can't just be nowhere. And in this oxidative phosphorylation, oxygen is reduced and the end of the electron transport chain is oxidized. That's where it's lost from. So like you do not have to memorize what's oxidized and what's reduced if you understand what's happening. It's like if you understand going from NADH to NAD plus means you're losing electrons, it's losing negativeness, then you can just automatically know loss is oil, so oxidation. If you know that water's being split and extracting the electrons in a photosynthesis, then you'll know electrons are being taken away, so that's oxidized. If you gains those electrons at the end of the ETC, gain is reduction, so you know that the oxygen is being reduced.
Stop!
Is that a question? Was that a child?
She unmuted.
She unmuted you. Am I muted? No, I'm not. Okay. This kind of thing, to be able to read this, mastering does a good job on that. You require it to get from product reactants to products. So basically, the enzyme makes the system unstable in some way or orients the system in a way to where the reaction is more likely to happen. That's this bottom one. So let's see. Specifics.
Can you go back 2 slides really quickly? I'm gonna
Sure. 2 slides on
uh,
this thing, the PowerPoint, or on my drawings?
on the PowerPoint.
Okay. Tell me when to stop. What are we looking for? I can say that.
Hold on. I don't know. It just looked very, very thorough. It had a bunch of little
This
diagrams
thing?
on it. That, and there was one, I don't know where it went.
This?
Yes, that one. That one.
Okay. So sometimes in class, I have everybody divvy up the stuff and everyone has to do different things, like different groups. So one group's job is to tell us the 2 types of energy, examples, biologically relevant. So basically what I did is I took chapter 6 and I broke it down into each section. Yes, this is chapter 6 stuff.
Okay, so it's not going to be on the test.
I mean, this is just chapter 6. This is all the stuff that chapter 6 is about.
Got it.
So it's, in a way, it's kind of like learning objectives, like on a more, on a detailed level where it's, like if you understood this, that means you know chapter 6. Yeah, so let's define metabolism. So can you do that? What's a metabolic pathway? 2 types of metabolism.
I got it. I didn't mean to interrupt.
No, this is why I'm here. I'm not here to, I mean, mostly I wanted to just do things that you guys would think were helpful. Please, this is good. What I'm looking for. Let's see. Okay, and then I'm gonna have to go soon because we all have stuff. So just to remind you, I have the keys for lab 8, which is the one where you did the sketches and you did the a couple of those questions about cell cycle. And then for that click and learn that you did, that particularly emphasized tumor suppressor, proto-oncogene, oncogene, that kind of stuff. So there's keys for that are open. So you do have to write about cancer. Either, there's 2 different options, I think, and I believe I have those as like possible essays. But just making sure that you go to the lecture and get the actual real definition. And this is based off of the click and learn, which is based off of like modern information. So it's not just dividing too much. It's also that they don't die enough, the cells. It's also, and possibly that they don't differentiate. That's from the one where you had the intestine and the cells didn't, they're supposed to specialize as they go up the villus. If you remember that animation, that they go up the villus and they're supposed to get specialized, but if they don't, then they can't move on, and so they just pile up. So basically the cells stop specializing. And then also they have to invade tissues to be considered cancer. So that doesn't have to be metastasized, but at least has to invade the surrounding tissue. And so that's a basic definition. It should be in the cancer notes. And we're looking at benign versus malignant and the process of metastasis. So these definitions here. I specifically tell you of a process of metastatic tumor is the secondary tumors, the ones that are from the initial tumor, and the cells end up in another part of the body and grow into a new one. So the metastatic process is actually very complicated in the sense that the cancer cells have to detach, which is weird for them to do. Not die after they detach, also weird. Be able to go through the bloodstream or lymphatic system and and not die there, also weird. Usually cells have to be attached to something, unless it's like you're a red blood cell or a white blood cell. The regular cells are not supposed to be in liquid and be able to exist or survive. Then it has to get to another spot, and then it has to actually have stem cell-like properties to where it can grow into a whole new tumor. It's not just about getting out there. Then it has to have the ability to make a whole new tumor. And there's a lot of things that have to go on in order for cancer actually to become metastatic and for metastasis to completely occur. So, and then in the metastatic tumor, it's still called just whatever initial cancer that the primary tumor was in. So like if this is breast cancer, then she might have a metastatic tumor in her lung, let's say, but that's not lung cancer. It's just breast cancer that's metastasized to the lung. And so cancers have different signatures, which is actually why it would matter, because if it's breast cancer, there's certain signatures you look at and certain drugs that kind of correlate with different problems. So it will be important to know what problems the cells have so that you use the right drugs and other therapies to target that kind of issue. And then here's a nice examples of, I'll ask you about like what are unusual things about cancer cells. And so all these are different ideas here, besides growing too much. So more specifically, that they can grow without getting signals to grow. When they get signals to stop, they don't stop. We've heard a lot about this immune system one recently because we're trying to help program the immune system to better recognize the cancer cells. It's like one of the strategies of immunotherapy, but cancer cells normally are not recognized by the immune system, which is weird. Angiogenesis means that they recruit blood vessels, which bring nutrients to them. So basically, like they make blood vessels grow towards them. They don't die. Cancer cells are also really weird because like you can basically cut off their oxygen it's fine. The mitochondria do weird stuff. Like they can do cellular respiration type processes and fermentation type processes in environments that normal cells would not be able to do, like human cells. So that's what this deregulating cellular energetics means, and basically means they're able to make enough ATP even without the normal types of oxygen and requirements. Invasion and metastasis, so cells are not supposed to be able to crawl around and leave and detach from other cells. Most of them are not. I mean, like white blood cells move around, but very few cells in the body do that. They're mostly supposed to stay where they're at. And remember, when you're talking about cancer, try not to anthropomorphize too much. And by that, I mean, the cancer wants to do that and it tries to do this and like all that kind of stuff. Because remember, the cancer cells are just behaving in the way that their molecules allow them to. And how does that happen? Their genetics code for certain proteins with certain shapes that can do certain things or don't do certain things. And then that makes the cell do certain stuff. And then they're cancerous or not, malignant, benign, make tumors, don't, all that stuff. Metastasize or they don't. So just the cancers are not like the cells are not having goals of getting somewhere and doing something. They don't have like a seeking a mechanism in the sense that it's not like trying with each other to spread or anything. Let's also remember too that cancer cells don't turn other cells into cancer. So it's not like a tag your it thing. They can change the cell behavior around them. Sometimes the cancer cells will end up making cells around them support their life instead of like really doing what they're supposed to. the blood vessel cells towards them and put out inflammation factors, even though they're not normally supposed to do that, the other cells, which then promote growth and stuff. So, but they're not going to turn other cells cancerous. That's a common misconception. Where the cells just, cancer cells basically crowding out the normal cells, and so the body processes just can't happen anymore like they're supposed to. And also you want to make sure you don't have to know all this stuff. This is all pretty much click and learn. Where's my thing? Here. So you're affected by cancer, and it's not an infection. So infections are like where you have other organisms that are inside of your body. This is not the case. This is just a aberrant version of your cells that are doing this. So you wouldn't say, you can get a viral infection though, and end up with cancer from that. It's like HPV. Most HPV strains, the virus don't do that, but some carry oncogenes with them. In the viral genome, there's an oncogene, and that can insert into your DNA. So now all of a sudden you have a version of a gene that makes a protein that makes the cells divide too much. Or sometimes, like with Epstein-Barr virus, which is what caused mono, I believe usually the reason for that, sometimes leading to lymphoma, is because the virus can insert viral DNA into the genome, and if it inserts it in a spot where it messes up genes that are needed for cycle regulation, then it can lead to tumor formation. So infections can lead to certain cancers, but cancer itself is not infectious. And you won't pass the cancer on directly because it's not an infection, but you could pass a virus on to somebody else, and that could lead to cancer development. That's why we all get the HPV vaccines now, use condoms to prevent HPV spreading. Giving women are the ones that have the consequences of it. Obviously, men are infected with it as well, but cervical cancer. So that's kind of that stuff. Cell cycle stages. Any other questions?
I'm good.
Okay. I can only see two of you. So
I am good as well. I had a quick question. I'm sure you've gotten it from a lot of people, but about our Lab 7 formal reports. I'm really anxious about getting my grade back on that.
well, I can't say that'll be super soon. Partially because like I gotta make sure you have the material and you have the labs to do and all this stuff to work on, you know, because if you don't have it, I can't expect you to do it, learn it. Good news is that, I mean, everything's in the book to a large degree as far as material goes in the mass training and whatnot, but obviously with lab stuff, I have to make sure that that's together for you. And yeah, so I've been working on that in another class as well. And of course, I want to have exams graded. 30 points versus 100 points. So sometimes I weigh that as well. So yeah, I mean, I know it's there. I don't have any kind of date that I can estimate for that at this point.
Okay.
Okay.
Well, I know, like, in my case, I'm really looking at that versus the drop date because frankly that's what it's come to for me.
So I think that it's going to be more important for you to get your exam grade back though before Friday. Because the 100
Yeah.
points versus 30 points, right?
Yeah.
So that'll be my goal for that.
Oh, I have a question. It's may just be me, but in mastering biology, I have no modules like upcoming.
Yeah, for the genetics.
Yeah, I have no. There's nothing
So
like.
they'll open tonight, presumably because people will take the exam tomorrow. Like they should. So that,
And is the exam going to be open for just one day?
yeah,
Is that,
they'll open at 12:01 tonight and be open till 11:59 tomorrow. I mean, yeah. So 12:01 meeting tomorrow morning, all the way till tomorrow night.
okay.
Last question, I promise, for extra credit, because at this point I'm just like fishing for as many points. And I know a lot of the opportunities that were going to be available are no longer available.
Yeah, so I just looked actually on this on the Houston Museum of Natural Sciences website this weekend. Kind of found some stuff that maybe they're streaming talks and things, like they're doing stuff along the lines of that, blah, blah, blah. Checking around. There's also things like I know there are places doing things. So the thought, first of all, there are a lot of lectures online already for that I have up for you that you can do those online lectures. So they're called like online talks in the bonus. And they're from previous speakers at Lone Star Kingwood. And so you can watch, they're all related to science, and you can watch their things and do a write-up on them. So that's an option that's there.
How long would you want those write-ups to be, or does it matter? What should they consist of?
Yeah, I mean, half page.
Half page?
Maybe yeah, I mean, maybe a page. I mean, mostly I need to know that you watched it and you paid attention to it and thought about it. And and thought about how it connects to what we talk about in class. That's what I'm looking for. So like more than 3 sentences, but don't go crazy. You don't need to use your time that way.
Do I get extra credit for gardening?
Gardening?
Through
I do have some stuff though, some genetics things that are extra credit that are like, if you do them, you can get the extra credit on them kind of things. As far as labs go too, I'm trying to narrow down to the few labs that I think they're like sort of a longer period of time kind of simulations or labs. So I'm going to get those out, and then you will work on them for a week or 2 weeks, something, maybe some till the end of the semester kind of thing. So that's my most recent plan for that, to try to put you in a situation where you can reasonably have time to do all the different things. Yeah. It'll be a lot less than when you normally have in class, but that's okay.
That is okay.
Everyone's okay with that? Yeah. No. Okay. Well, I'm gonna go then.
All right.
Thanks for the study sesh.
Yeah.
Sir.
Good luck. Get some sleep tonight. I know sometimes that's counterintuitive when you want to make sure you have time to study, but your brain works better when you're rested a reasonable amount. Yeah, and don't wait till the very last minute to do the exam. It'll be way stressful. I don't want that for you. So give yourself at least 3 hours. And, if there's any chance that something's going to go wrong later in the day, just do it in the morning. You can have a sheet of paper. You just have to show me it's blank. So you can work on that. And in fact, I would have a sheet of paper because then you can write down your cycle on it. And that way you can just refer to that as you're doing it instead of having to think every single time about the answers. If you haven't done the practice exam to check your webcam and make sure everything's good to go, you need to do that like much sooner than later because you'll need to contact tech and see what needs to be changed if something's not going right. Most people have done that though. Okay. Everything went great for my class that took their stuff. What was it? Friday? Yeah, one of my other classes took stuff Friday. All the web stuff went great. So, and like I said, I've done this 2 or 3 semesters with the online testing, and it's been fine. People like it actually because you're kind of in a quiet place. You get to sort of take your time. They're not trying to do lab afterwards. It has advantages. Okay. All right, guys.
Bye, guys.
Have a good night.
Bye, everyone.
I'll see you later.
Bye.
Bye.
Hello.
They didn't ask for no fucking Chinese food, or I don't want to got some Chinese